at the targeted therapies meeting, I was given the other topic, and we joked at the meeting there were 7 topics, and the 8th was other in most of the categories. So I chose as other immunomodulatory agents, I chose IL-2 and focused my discussions on that molecule because it has shown us great promise, but yet has been associated with toxicity in the past. High rates of grade 3 and 4 toxicity in wild type IL-2 that's approved for kidney and melanoma. So I reviewed the immunobiology of the IL-2 receptor that explains some of why those toxicities result, and the efforts to develop either non-alpha IL-2 agents or alpha-beta IL-2 agents that bind to the trimeric receptor as opportunities to enhance the efficacy by potentially downplaying some of the toxicity. So one of the compounds I talked about was STK012, made by Synthekine. It's an alpha-beta IL-2 mutein. It binds to the trimeric receptor but spares the gamma subunit. And so they think that not only spares the upregulation of T regulatory cells within the microenvironment, but also diminishes the expansion of natural killer cells in the periphery, which has been linked with vascular leak syndrome. I think we'll have to see these drugs in the clinic. Their phase 1 study has been ongoing for a while yet. No clinical data, but we've been pleasantly surprised here at Sarah Cannon with the patients that we've treated. I also uncovered a bispecific made by Roche that targets PD-1 and IL-2 in my research, and that was interesting to me because the confirmation of the bispecific seems to be key to its ability to activate anti-tumor specific T cells and to spare some of the unwanted side effects like T regulatory cell stimulation. So we'll see that if that's in the clinic. It's just barely entered trials, and so we'll wait for more results there. But at a meeting in 2023 where we were discussing all of the advancements essentially over the last 3 years, because the last time we had that meeting in person was in 2020, right before the COVID pandemic began, I could see a little bit more movement in the drugs that had been developed. Now, some others decided the cup was half empty, that we've evaluated a lot of therapies, but we haven't made any progress. But I think I walked away from the session that I was a part of, in agreement with the panelists that we were lucky to identify PD-1 and PD-L1, and it's probably going to be more difficult to make that next step. And it's probably going to be less related to the PD-1 pathway than many of the therapies have been that have just built upon the PD-1 backbone. That's why I liked IL-2, because it was different enough that I could see it making a difference.